in there that we have studied, and there's magnetic memories in there that we will study, all in the ICF. So go back there and just look at it. If you want to know the names, turn them over and look. I'm going to put in the tennis. We're going to start down there. A non-competitive inhibitor or competitive?
Competitive.
What is it competing with?
Maltose.
Maltose. So we have maltose here bonded to what? Hexokinase. Try again.
Maltase.
Maltase, right? So, mystery molecule is competing with
substrate.
the substrate for what? What are they competing for?
The active site.
The active site. Is this molecule competing for hexokinase's active site?
No.
So is it a competitive or a non-competitive inhibitor?
Non-competitive.
Non-competitive inhibitor. What does the word inhibit mean?
It's like it's moving in.
Inhibit.
To limit or control?
To limit, to control, in this case to stop. Why is hexokinase being stopped right now? How is it being stopped from functioning? James?
Because there's a surplus of fructose 6-phosphate.
Not exactly. Hi, Sky.
It's closed off the active site so that the glucose and the ATP can't get in.
Right. Okay, active site is closed off. What's closed off here? Active site. Okay. Rock and roll. Let's go up to the front. You ready? Daniel, you ready to go, buddy?
Yep.
Okay, we're going to think about a whole bunch of things. Why? Because, hey, it's fun. First of all, it's fun. Second of all, it's fun. All right, let's go back in time. What did I ask you to think about yesterday?
Why we would use the other hexokinase in our cell, why we would turn them on.
Correct. But from an ancestor point of view, what are you trying to place your mind in? Yes, sir.
How the mechanism helps our ancestors.
Exactly. So we talked about our ancestors, but how far back do we go in time?
2 million.
Yeah, I picked 2 million years. So this was before Homo sapiens were on the planet, right? So, from an evolutionary point of view, where were our ancestors living 2 million years ago?
I'd say a very dangerous place.
Very dangerous place, good. And where was that dangerous place? Could have been a lot of places on the planet that would have been dangerous for our ancestors, but they were in one area of the planet, Africa, right? Different parts. So, what I want you to think about is, how did this mechanism here, this is a mechanism that we're going to look at in many different ways. Here, how does a mechanism of having enzymes turned on and off, how did that help our ancestors survive? Okay? That's what we're trying to figure out here today. Because, squad, look. Here's a key, right? You could take this key and open the classroom door. But when you do that, you're using your amazing brain. You've heard me say this before. That's consciousness. You're aware that this key fits in room 114. The other thing I'm trying to get you to think about at the same time is, how does this system work? How is it regulated? These enzymes aren't talking to each other like we would, but there is a communication system that's quite sophisticated, but there's not a consciousness in there. So what was the word that we talked about yesterday that begins with the letter R?
Regulation.
Yeah, how is this system regulated? How is it controlled? Right? It's not like a teacher in the classroom saying, okay, you're out of uniform. I'm controlling that. Right? So this is what you're thinking about. How does this system work? Like, how's it regulated? How did this system, and hey, don't forget, liver cells and fat cells, how did this system allow our ancestors to survive? As James mentioned, in very dangerous environments. Okay, let's fire it up. Before we jump in there, answer me this question. Okay, one of our ancestors came across some fruit. He or she ate it, put a whole bunch of glucose in the blood, was sitting down under a tree like you're sitting down now, right? Glucose, what's the mechanism here of the spin move? Facilitate diffusion, right? So glucose enters this cell. In this picture here, what is not going to happen to that glucose molecule?
It's not going to go through glycolysis.
Okay, why? What will not happen to it right away?
It won't bind hexokinase.
And it will not be what?
Diffused out.
It's not going to be phosphorylated, right? Why is it not going to be phosphorylated?
Because there are no available hexokinases.
Okay, so you're jotting this down. So first of all, we're thinking now, told you we're going to do a number of different things. Let's just call him Ancestor Ernie. Ate some carbohydrates. He's resting under a tree just like you're resting now in your chair. The glucose diffuses into the cell, but the energy demand is not there for glucose to go through what set of reactions, Sky? What set of reactions that we just studied that glucose goes through, Carly? Glycolysis, correct. Cellular respiration, right? Yeah, you could use either of those two words. Okay, what do you remember about when glucose, when my finger's pointing to now, when glucose is phosphorylated, it becomes glucose 6-phosphate. What does this do to that glucose molecule? What would it allow it not to do?
Can't chemically diffuse out of the cell.
That's right. What's going to happen to this glucose, and this glucose, and this glucose? James?
Diffuse right back out.
They're going to diffuse back out because they're not phosphorylated. All right? Now remember where you're at, squad. These are molecules that came from some fruit. Look where they're going. They're going to a muscle cell. The energy demand is low. All right, let's take a bunch of the glucose here, not all of it. This glucose, by the way, could do that same thing in all kinds of cells in different muscles. Energy demand is low. All right, when that glucose enters a fat cell, squad, what's going to happen? What's going to happen? Yes.
It's going to be converted into
To-
stored energy.
Good. Perfect. Beautiful. So, if you're not aware of this, you're aware of it now. Fat cells do take glucose, convert them to, what's this molecule here, Joanna? Convert it to lipids, right? Now, good work, squad. You're on fire as usual. I don't know what I'm going to do, Mark. Right? I'm going to wake up Tuesday or Monday, and it's going to be like 1 o'clock. I'm going to, I'm supposed to teach. Where's everybody? I'm going to call an impromptu meeting. Okay, you ready? Hey, I got an idea. Shut the tape off. Okay. Let's go over this. This came up this morning. This is still not as clear as I would like it to be, right? Let's review the facts of the case. Sarah, true or false there's stored energy in this molecule?
True.
True or false there's stored energy in this molecule?
True.
True. Can the cell use the stored energy in glucose to do work,
Not easily.
not directly, right? What form is the stored energy in? Electrons, right? Okay. One thing I want you to be clear on here, look, squad, when ATP is produced, it is used immediately. Your cells don't take ATP and stick it in a freezer and use it and store it up, okay? So when we talk about stored energy in biology, we're talking about lipids. And let's take some of these glucoses here, bring them up to the liver. When the liver bonds them together in these chains, right? What do we get? What's this molecule called? Yeah, it looks like cellulose, but notice there's no,
Glycogen.
these are all alpha glucoses. And there's a bond here. This is glycogen, right? So, that's scary. Glycogen, stored energy. Lipid, stored energy. Yeah. Told you we're doing a number of things. Think now for a minute. Stay on this path of stored energy. How did this mechanism here of liver storing glycogen and fat cells converting sugar to lipids stored, how did that help us survive? And this mechanism is obviously still in our bodies today. Yes?
It stored the energy until we would need it.
Good. And why was this critical?
Because we might have not needed it at the time, but when something came along and we needed that boost of energy, and then
Close. Sky?
Would it have to do something with like famines? And, like,
Famines, yeah. But so jot this down. This mechanism here, this pathway, we did not eat 3 meals a day whenever we felt like it. So the food, the availability of food was completely different, right? So we developed these cool mechanisms of storing energy that we ate so we could use it when we what?
Needed it.
Yeah, when we weren't eating regularly, right? So one thing you may not be aware of, squad, you are burning right now. When I say burning, you're producing a lot of ATP because the lipids are going through cellular respiration. The lipids are coming from fat cells. It's a normal pathway. Okay, any questions on that? Okay, let's do what we normally do, a quick 22-second review. We are building a monster picture of one muscle cell at rest that represents your body right now. You're sitting down, and one of our ancestors' body muscle cells 2 minutes ago sitting under a tree. Right? He or she ate some sugar. The sugar got stored as glycogen for lipids. Right? He or she is producing 6 ATP per second, and that's enough ATP to keep this muscle cell functioning at rest. Got it? We're good. Okay, new question. The calmness of the day is about to get crazy. Because what is now coming at our ancestor who was comfortable under the tree? Yes, the old saber-toothed tiger. Okay? So, jot down our ancestors being attacked. Right? So what I want you to figure out is, I want you to build me a picture of what would happen anaerobically. What anaerobic changes would take place. What are we talking about? Glycolysis now. Tell me how many ATP are going to be made. So I want to know what the changes are. I want to know how many ATP are going to be made. And I want to know why this system, why this mechanism, which is increase this person's ATP. How did that help him or her survive the saber-toothed tiger attack? Told you this was fun. Anybody? Okay, Paulo, go ahead, buddy.
Well, first, all the hexokinases are going to open, so the active sites can take glucose.
Okay, so he says first the hexokinases are going to open. That is correct. Let's stop there. How do they open?
Well, the glucose 1,6 or fructose is going to leave the allosteric sites, so
How? Why? You're definitely right. He said correctly, look, we'll model it like this. He said glucose 6-phosphate will leave hexokinase allosteric site, correct. Hexokinase will open, right? Clearly, there's now more glucose that can enter the cell, right? Oh, I don't like when that happens.
It's like me trying to like fit all over again.
Like you what, Charlie?
It's like me trying to like those magnets all over again.
Ah, you're saying, sir, deal with it. Okay, right? Now here's my question. This is correct. Hexokinase is open. We're going to get a phosphorylation of glucose. What removed glucose 6-phosphate from that allosteric site? How'd that happen?
The demand for ATP went up, so enzyme 2 now
Ah,
comes in.
beautiful.
And needs glucose 6-phosphate in order to make more ATP.
Here's my baby enzyme 2. So enzyme 2s, we're jotting this down, are going to remove glucose 6-phosphate. So what is think of it like this, squad. Glucose 6-phosphate is an off switch for what molecule? Hexokinase. What's the on switch for hexokinase? So now we're talking about biological switches, right?
The enzyme 2.
Enzyme 2, right? Got it? I think you got it. Okay. I want you to envision that all of these hexokinases are turned on. This is a life or death situation we're painting here, right? Joanna, did you work out how many ATP could be produced? Pardon? She says 10 ATP. Can you tell me you're on the right track? How'd you calculate that? Okay, there's 10 hexokinases, which represent 10 simultaneous glycolysis reactions going on. So just imagine 8 enzymes here, and then another 8, and another 8, right? So there's sets. Glycolysis, think of it as a set of 10. How many ATP are produced with 3? So how many would produce with 10? Close.
20.
Yeah, 20, because there's 2, Joanna, you know this, there's 2 ATP net, ATP produced. So we're talking about 1 second, right? So we've gone from 6 to 20. Have we increased our, hi, you okay, buddy?
Yeah, hello.
Okay.
Can I have all the chairs in the back of the room tonight before you guys go home?
Absolutely.
Thank you very much.
Anything else?
Nope, that's
Okay.
everything.
Merry
Have a good
Christmas.
March break.
Okay, have we increased our ATP production? Did we do it, did this mechanism do it incredibly quick? Was that beneficial? Why? Why is that beneficial? Go back to the saber-toothed tiger, Daniel. Why is this beneficial?
Because we need the energy to run away from the tiger.
Yeah, it's pretty basic, exactly. We need the energy to run away from the tiger. Why do we need anaerobic energy to run away from the tiger? I'm not talking about aerobic energy.
Because it's going to be hard to breathe.
It's going to be hard to breathe, and it's a sprint to maybe the cave, over the rock, whatever. We're talking short distances. You with me? Okay, what do you think of this system? True or false here? Hexokinase is a protein. Okay. Does anybody have any idea of how, we haven't studied this yet. Do you know how these proteins get made? Okay, that's a big story we're going to learn. Here's what I want you to think about. Imagine these ancestors that said, look, why would we make 10 hexokinases when we only needed 3 at rest? They're going to say waste of time and energy. So when they came around that saber-toothed tiger, this is an incredibly efficient system. Here's a simple model of DNA. So here's this person sitting under the tree eating the apple. Only got 3 hexokinases. The tiger comes at him. Message, without even him thinking, goes from the brain. Sky, you with me? Goes from the brain to the muscle cells. Oh, tiger coming after me. You will learn there's a hexokinase gene, right? That gene gets turned on, and the cell makes new hexokinases. It's an incredible system, but you know what? Even at the best of time, it takes 5 minutes to make the new hexokinases. So here's the speed of the guy running away. That's it. What happens? Lunch to Rooski. It's over, right? Okay. This ancestor they went extinct. Okay? Didn't work. This backup energy system, let's jot that down. This is language you're going to hear me use after March break. But yeah, after March break, because we've got 20 minutes left. Okay, this is backup. What I'm teaching you here today, backup energy system 1. Specifically, backup anaerobic energy system 1. Okay. So backup energy system 1 increased ATP production how much? Just over 3 times, right?
Sorry, increased ATP production?
Yeah. And this is, think of this as a escape velocity, right? Okay. Now, I'm going to start teaching you backup energy system 2. We can get more ATP out of these systems than what we just went over. You with me? So this protein I'm going to put on the board, which some of you looked at the back. Do you remember reading anything at the back of the names of anything? Did you turn them over? What did you read? Creatine. Creatine, anything else? We'll study creatine after March break. Okay, here's this enzyme. It's very cool. I want you to understand. Look, here's what I'm going to do. This enzyme, I'm blowing it up, right? I'm going to move it in a minute. This enzyme is in the ICF, okay? Right? So we take this enzyme out. I'm going to put it here. You sketch it. Just did, I apologize. I'm shutting it off.
Did you want us to try to shut off?
I do. Yeah, I just changed my mind.
Okay.
Yeah. Let me get rid of these. Okay. All right. Anybody doing anything exciting other than sleeping and eating? Huh? Movies? Do you have any movie fans in the audience?
Oh, yeah.
Okay. Gotta love movies. It's almost good as class. Here we go. Sir, are you kidding? No, no. Okay. So, let's identify a couple things. Right here, squat. This, bye-bye chalk. Okay, this is a very, very cool enzyme. Allosteric site. Allosteric site. Okay. Enzyme is shut off. I'm going to put an ATP molecule in this allosteric site here. So imagine that, eh? Now think about this, squat. I'll name this enzyme in a minute. This enzyme is in the anaerobic pool. This enzyme is not part of glycolysis. You have not studied this enzyme before, you're being introduced to it right now. But you're asking yourself, there's an ATP molecule bonded to it. That shuts it off. What does that tell you? What does it tell you? Column?
That if ATP is needed, the ATP on this allosteric site comes off.
Yeah, so what would it tell you now?
That it's going to activate. The enzyme's going to activate the.
Yeah, no, ATP, when ATP is in this allosteric site, the enzyme stays off.
Yeah, and then whenever we use it, opens up.
Right. But the fact that it's ATP is bonded to that allosteric site, would you say the energy demands of the cell are high or low?
Low.
They're low. Clearly, if they're, I mean, you don't take the only usable energy, right? So, in this situation, low. Let's follow his lead. We'll get to this allosteric site later. ATP will be removed. We'll talk about that in a minute. When this enzyme opens up, NADH I need a proton. NADH will bond to one active site here. And check this out. This is where we have to do some serious thinking right now. Boom. It's a double bond. What do we got here? What do we have here?
Pyruvate.
Pyruvate. So, simple language, LPH. So the name of this enzyme is LPH. We've talked about a couple of active sites. We have pyruvate right here. Coenzyme, Daniel.
Isn't LDH except for cholesterol?
Close, you're thinking of
LDL.
HDL or LDL, right? Now we're going to get into something that some of you are very familiar with. LDH is short for lactase dehydrogenase. So remember, this is an anaerobic reaction, squad. All right, let's check this out. Are you ready to start the reaction? Let me tell you one other thing. Not only is this enzyme anaerobic, right? The cell is an oxygen debt. So let's walk over here for a second. James, follow me. We know that this system that I don't have here, we could go to the back of the room in a minute, which we will. What's the name of the enzyme that pulls the electrons out of the electron transport chain?
Oxygen.
The oxidase complex. Oxygen debt. There's no oxygen. Why? What is our ancestor doing right now in this story? Running
Really fast.
really fast, right? Okay? Can't get oxygen into the mitochondria.
Excuse me for the interruption. If there are any grade 12
Do we not get to go to grad if we don't hand those in?
You'll get them in. It's only March. Okay, let's back up here. Let's do the reaction. Here's what happens. This cool enzyme here will swing, boom. When those two molecules bond to their active site, molecule swings. Check it out. The reaction is... Carly, you're intrigued.
Sorry. This is my interesting phase. Carry on.
That's okay. The reaction is straightforward. What's intriguing is why. But we'll do the reaction first. Okay, look. Dehydrogenase. So the hydrogen and electrons, this is going to be a redox reaction. It's going to be transferred. See the double bond here? Look, carbon 1, 2, 3, the middle carbon. Double bond is broken. Right? Look what we had here, squad. Now, that hydrogen and electron are bonded to oxygen, right? This electron is going to bond to carbon. What do you think will be attracted to this electron? A what, James?
A proton?
A proton, exactly. So there are these protons hanging around. Boom. Now we have a new molecule. All we did is convert carbon to OH, H was a double bond. Yes.
So this is just a proton that was around the cell.
Yeah, there'd be a proton that would bond to an active site in there and
Okay.
LDH. Okay.
Yes.
Okay. Let's look at the after picture here, squad. I bring this out. NAD plus. Now, move this over here. Here's a molecule you've heard a lot about. And does anyone know the name of it? Right here. It's not pyruvate anymore. Yes, sir.
Lactic acid.
Good. Call this lactate. So, the product of this reaction, anaerobic. So think about a couple things. Lactate is produced in an NAD plus. Yes, sir.
So there's now only one double bond left, an oxygen.
Correct. Yep, correct.
And is the enzyme stays open?
Yes, I'm going to keep it open because it would conduct another reaction. We're going to go to the back of the room and try to figure this out together. Why? Because it's fun. Okay, so you're, as usual, you're doing good work. True or false, Guy? First of all, we got a little bit of ATP here out of this reaction. Can this ATP be used to phosphorylate muscle cells to help run away from the saber-toothed tiger? Yes. Do you see any ATP here? So there's a little bit of ATP production here, one, right? Let's go to the back of the room and figure out how this is beneficial. Okay, let's go. Come on over here. Okay, we'll spend more time on this after March break. When I put up on the board, up on the blackboard, oxygen debt, if you're in debt, it means you got, you could interpret that one way, you got no money. It doesn't necessarily mean you have no money. You could owe somebody, you could be in debt to somebody, right? But in this case, oxygen debt is specifically, there's no oxygen in the mitochondria, right? So you're going to just listen and make a note later. Tell me to stop when you're confused. If there's no oxygen in the mitochondria, will the electrons get removed from the electron transport chain?
No.
No. Will all the coenzymes eventually be saturated, full of electrons?
Yes.
Because they can't deliver them to the electron transport chain.
Yes.
This is what's happening when the person is running away from the saber-toothed tiger. This is what happens to anybody who runs or skates or does anything intense and fast. They can only hold that for a short period of time. Doesn't matter who. You can take Bolt, right? World's fastest man. He cannot run at top speed for more than 15 seconds, if that. If he, right? Because his whole system is shut down. He just happens to get a whole lot further than the rest of us in those 15 seconds. Yeah, no kidding. Okay, this is the situation we're in. No ATP production, electron transport chain, no ATP production in the Krebs cycle. Let's go back and see what happens. Sorry. Okay. So here is the after picture, right?
Sorry.
Let's put it up head to head with what's going on in glycolysis. Right? Okay, follow me. Glycolysis, and I put these in order. Enzyme 1, 2, 3. 4, Splitterowski. 5, does 5 conduct a redox reaction? Yeah, with what?
NADH.
With what?
NADH.
NAD plus. Right? Okay? Now, let's think about NADH's glycolysis. They diffuse into the what?
Mitochondria.
Mitochondria. They do a switcherooski with what?
FAD.
Will there be no FAD available in oxygen debt?
Yes.
Mm-hmm. Will all the FADs get full of electrons?
Yes.
Yes.
Okay, let's do that. So, let's just put eventually, right? Look. Eventually there's no fads, right? They all become FADH2s. So, where did this NADH come from?
Glycolysis.
It cannot do what?
Diffuse.
All right. Now, if we follow this, enzyme what produces ATP? 6. Enzyme 7, switcherooski. Enzyme 8 makes what? ATP. But you go above those 3 penguins to here, right? The cell needs NAD plus. If there is no NAD plus available, will enzyme 6 phosphorylate? No, the next reaction will not happen. So check it out. This NADH, water's bouncing that around, boom, it's going to bond with the now available LDH. Switcherooski, what's now available to bond to enzyme 5? NAD plus. So that cell can make anaerobic what? ATP. Good. And thank you. That is the purpose of this reaction, right? this enzyme is freeing up NAD+, so 6 and 8 can make more ATP during oxygen death. Got it? Okay, let's go make a note on that right now.